Good afternoon and welcome, ladies and gentlemen, to the La Jolla Institute for Immunology. I am Erica Ollmann Saphire. I am a research professor and also the president of the Institute. I would like to give you today a look inside our laboratories. So this is the institute that you're looking inside right now, La Jolla Institute for Immunology. We are something special. We are an independent, nonprofit biomedical research institute. We're a think tank, and we focus on the human immune system that keeps you alive and well in response to infectious disease, cancer, heart disease, neurodegenerative disease. In that area, we are among the top 5 in the world, and we are a 501c3 tax-exempt charitable organization where your donations fund our research. Our faculty here study every aspect of the immune system, from its fundamental workings to how we defend every disease, we like to say, from A to Z, from Alzheimer's to Zika. And our faculty are on a mission, and this is our mission here. The Institute will engage in a world-class biomedical research program with a focus on the immune system. And the reason we focus on the immune system is because of its singular power, beauty, and precision to keep you alive and well and coordinate all the other systems in your body. The research that we conduct, share, and partner will be such that the results of our discovery program will make outsized contributions to the betterment of the human health. And that's an important point, that we aim to make outsized contributions, because our staff, our focused staff, 24 top-notch faculty, 500 total scientists, including 200 trainees, very much punch above their weight, with their research noted and described and discovered and analyzed and highlighted by all of the major news networks nationally and internationally. And some of our research, for example, in COVID, has set national and international policy, and our scientists serve on international task forces for how the world should respond to certain infectious diseases. Our scientists are divided into 3 research centers. We work on autoimmunity and inflammation. We work on infectious disease and development of long-lasting vaccines. And we work on cancer immunotherapy and a lot of other viruses, like we say, from A to Z. Today, we're going to hear from one of our research faculty, somebody who's very special, Professor Mitch Kronenberg. Now, in addition to being one of our research faculty, Mitch was our former president. He led us for 18 years. And while he was our leader, he was voted by a local business journal as the most admired CEO. Under Mitch's leadership, the size of our faculty doubled while our operating budget tripled, and we forged important new alliances for new frontiers. Now, Mitch himself, as a scholar, has authored more than 500 different scholarly articles, and he was elected as a distinguished fellow of the American Association of Immunologists. His research uncovers how certain white blood cells, lymphocytes, operate on the front lines of immune defense, your innate immune system, how these cells patrol the barriers where your insides meet the outside world. We call these mucosal barriers, like your eyes and your mouth and your gut. Regulating and patrolling these inside-outside mucosal barriers and regulating all the complex activities that happen in those places is essential for defending you from lots of different pathogens, and coordinating all those activities is essential for controlling diseases like colitis. Now, Mitch's bachelor's degree is from Columbia University. He moved to the West Coast for his Ph.D. from Caltech. He led our institute for 18 years as president, and now I get to work side by side with him as our chief scientific officer. And I've often said that the greatest honor of my career is that I get an hour of Mitch Kronenberg's time every week. We are lucky today to hear from him in a unique capacity, not from his voice in administration and leadership of this institute, but from his own research laboratory and the discoveries that come from there. So we're going to turn the camera now to Mitch's laboratory office, where he's going to describe his research and discoveries at the mucosal barrier.
Yes. Thank you very much, Erica. That's a very kind introduction. And thank you everyone for attending and giving me the chance to share my excitement about studying the immune system and the aspects in particular that we and some of my colleagues concentrate on. I want to begin by giving you a little bit of a lesson about how the immune system works, the systemic immune system. Immune cells patrol your body. They're in the blood. They come out of the bone marrow, they're in the blood, and they enter into what are called lymph vessels. And the lymph vessels are these little green lines on this body. And so the lymphocytes are circulating in your body between the blood, into the lymph vessels, and back to the blood, looking for something to respond to. And all the little green circles here are what are called lymph nodes. That's the glands your mom squeezed in your throat. You have swollen glands, my daughter, my son, those are lymph nodes. And the lymphocytes go from the blood to the lymph vessels in the lymph node. They look around for something to react to. If nothing's there, they go back on the highway. You might think of the lymph nodes as a station, and they go back on the highway looking for something to respond to. And if they find something in the lymph node, then they might enter the tissues. And even within the lymph node, these cells are constantly moving around. You can see here, and particularly the red and blue ones are kind of attached to each other. The green ones are mutant, and they don't really know where to go. But the lymphocytes are looking around for something to respond to. And most of your body, most of your internal organs are sterile. What we've been studying for many years is immunity at the border or at the barrier surfaces, which really provides special challenges. And if you think of your body, I told you the inside is mostly sterile, but we, of course, connect with the outside world through our gastrointestinal tract, through our lungs, through our skin, through our reproductive tract, and so on. These are all barriers. And these are places where pathogens tend to enter, bacteria, viruses, protozoa, and so on. Our barriers have a lining. They have a surface. And these border cells, we call them epithelial cells. And we often draw them like this. It looks like a wall or a fence of some kind. However, that's not really the way it looks, because the surface is highly folded, as you can see on the right, in order to increase the surface for absorption of nutrients or for intake of air, etc. And so what this really looks like is not this rectilinear wall, if you will, but more something like this with many, many folds. In the intestine, we call these folds villi. And this gives you an enormous surface area. And if we stretched it out, the surface area of your gut or your lungs would approximate the area of a tennis court. It's really quite remarkable. Now, that barrier protects you, of course, but your immune system is also there guarding the barrier. And in fact, about 1 in 10 cells in the barrier are white blood cells of various types. So on the right here, what you can see is a fluorescent micrograph, a cross-section of several villi, and we mark the epithelial cells in red, and in green are all kinds of white blood cells that are sitting there on guard, either in the epithelial layer, in other words, in between the red cells, or just below it. So your immune system is there to guard the barriers. As you might imagine, the barriers are very important. In this post-COVID time, I think almost everyone has heard of antibodies and how antibodies are very important to protect you. And it turns out there are 5 kinds of antibodies that our body makes, and the drawings of their structures, the outline of these is shown here. And of these 5 kinds, IgA is the specialized antibody of the borders or the mucosal tissues. So it is in your tears, your saliva, breast milk, and the intestine, and so on. And your body makes a tremendous amount of IgA to protect you. It's about 15% of the antibody in the blood. But also, you're making an enormous amount in your secretions. In fact, in 1 month, we produce about 1/4 of a pound of IgA alone. And that's to illustrate what that, it doesn't all collect like that, of course. If we consider the barriers, the immune system is confronted with the outside world. We tell our students, or we used to tell our students, that a principal job of the immune system is to distinguish self from non-self. And I'm going to tell you that's not strictly true. But what is self? Well, when I look in the mirror, I see a handsome young man, and that's a bit delusional, but self includes a microbiome, and I'm sure many of you have heard about the microbiome, and I'd like to talk to you a little bit about it. It's estimated that in our human body, we have almost as many microbial cells as human cells. There's debate about the exact number, but there's near equivalence. But if we consider not cells, but genes, there are many more microbial genes than human genes. Each of our cells has an identical set of approximately 20,000 genes. But because we might have 1,000 types of bacteria, and these bacteria have, bacterial species, if you will, have different genes, we probably have millions of bacterial genes. Bacteria are very small, ancient cells that we call prokaryotes. But we also have other kinds of microbes, single-celled organisms that are yeasts, that are much larger. We don't have as many yeasts as bacteria, but yeasts are big, and they make a lot of stuff that can also influence us. And all these living organisms, just like we are subject to viruses. And there are viruses that live in our gut. Some of them are not very harmful. And there are also viruses, we call them bacteriophage, that live off the bacteria. So life is very diverse and very miraculous. And we ourselves are, in a way, an ecosystem. If you think of these organisms, most of the body's sterile. So they're confined to lung tissue or the interior of the gut or the skin. But they make stuff. And here's a metabolic diagram of what they make. And there won't be a quiz on this. Luckily, there's nothing for you to get from this, except metabolism is complex. Many of the compounds, we don't know exactly what they are. But these microbes are making stuff. And even though the microbes don't normally get into our body, the stuff they make can get all over the body. And that includes not only the bacteria, but the viruses and the yeasts as well. If we consider the different parts of the body, the colon has the most dense and diverse microbiome, and that's probably not surprising. The colon, like many organs, is subject to inflammatory diseases. Inflammatory bowel diseases are quite prevalent and increasing. It's estimated they affect about 1.6 million Americans, and most people are diagnosed before the age of 35. These are chronic, lifelong conditions, and they can be treated, and there are new biologic mechanisms coming along, but they cannot be cured. They have multiple causes, but one cause is believed to be a breakdown of the normal détente, the normal peaceful relationship we have between the microbiota and the resident immune system that resides in the tissues. There are really two kinds of inflammatory bowel disease. Crohn's disease, you can see on the left, which can affect both the colon and the small intestine. Ulcerative colitis is predominantly in the colon. And these two types of inflammation have different histologies. So the ulcerative colitis lesions tend to be more superficial, although they are more continuous, as symbolized in the diagram here, while the Crohn's disease lesions are deeper and have a big, almost granuloma-like formations, and they can even penetrate the wall, but they tend to skip areas. And in reality, these are two entities, but it's more of a gradient. There are forms of Crohn's disease which look a bit more like ulcerative colitis and so on. So there are unique challenges that face the immune system in the tissues. And one is they have a constant exposure to non-self materials, microbial materials, food antigens, things that are inhaled, things that might be on your skin. As a consequence, perhaps to cope with that challenge, there are specialized immune cells that reside there that are somewhat different from the circulation. I don't want to make that dichotomy too stark either, because circulating lymphocytes, after seeing antigens in the lymph nodes in those stations, can go and then reside in the tissues. But there are also specialized cells whose job is, from the beginning, to reside in the tissues and patrol the tissues. And so we have a complex resident immune system in the barriers that must be present and ready to respond, but controlled so that it doesn't cause unwanted reactions. So in essence, then, if we think about this concept of self and non-self, it's not true. What might be more accurate is what's dangerous. The immune system must respond to that, and it must tolerate and ignore those things that are not dangerous. That's the challenges of the barrier, if you will, of the barrier immune system. What about we investigators who are studying it? What are the challenges for La Jolla Institute investigators interested in barrier immunity? Well, one is just to understand the specialized border immune cells and their interactions with the microbiome, with food, or other agents that might penetrate the tissue. And years ago, when I started in this area, there were very few people investigating it, although now it's become a very active field, a beautiful field with lots of findings. We need to determine ways to effectively vaccinate the mucosal immune system, which is an area of active investigation I won't be able to talk about today. And we need to determine the causes of diseases, such as inflammatory bowel disease, food allergies, severe asthma, and many others, so we can effectively work for cures. And what's our toolkit to address these issues? Well, it's advanced techniques using immune assays, imaging, which we'll hear more about soon, later in the talk, genetics, and computational methods. And I'm going to tell you about a few types of work that we're doing. And during that, we're going to actually visit the laboratory. But before I launch into that, I do want to acknowledge the work of Mick Croft. And Mick has been working on lung inflammation and skin inflammation for many years. And one of Mick's findings has given rise to a treatment for severe atopic dermatitis, which is about to enter into a phase 3 clinical trial with brilliant phase 2 results. And we're all grateful for Mick's discoveries in that area. Okay, so I want to talk to you a little bit about model systems that we use to understand a very complicated problem. And one of the model systems we use is precision genetic surgery. And that means that if we consider that there are 20,000 genes present in all your cells, we can knock out, eliminate, or ablate one of those 20,000 genes by design. And we can even go further than that. We can say, I want to take away one of the genes, but only in a certain cell type. I want to take away the ability of a gene to make a protein, which is the only function of genes, the essential function of genes. They make proteins. I want to eliminate that ability in the barrier cells or in a type of white blood cell. And that's the kind of one of the advanced tools that we use in the lab. And it was invented by Mario Capecchi, who was awarded the Nobel Prize for his discovery in 2007. So here's an example of a precise genetic surgery that we did in my laboratory. And this is a micrograph of a cross-section of the intestine. The epithelial cells, those barrier cells in this micrograph are marked blue. And you can see that here. And underneath the dotted white line is deeper intestinal tissue. Now, our gene of interest is marked in red. And you can see it's expressed both in the epithelial cells on the far side of that dotted white line and deeper in the intestine. And if you look at the merged image here, the epithelial cells, which have our gene of interest, I won't even give you the name because it's not essential to illustrate the technique. You can see the epithelial cells are purple because they're blue and they have the gene of interest red. And what we did is we knocked out, if you will, the ability of this gene to make a protein, but only in the epithelial cells. So in the lower graph here, you can see that the epithelial cells still mark blue, and there's still red being made, red protein being made, but no longer in the epithelial cells, which now no longer look purple. So we knocked out a gene that's expressed, one of the 20,000 or one of the 12,000, since not all the genes are making proteins in every cell, but one of the genes expressed in these barrier cells. And what we want to do is ask, how did that affect the microbiome? And there was an effect. And one of the bacteria we are interested in is called SFB, segmented filamentous bacteria. And this bacterium has the ability, to a varying extents, to get through the mucus, which protects these barrier cells, and actually touch the barrier cells, while most of the bacteria are in the opening or the lumen of the intestine. Here's a beautiful micrograph that my colleague Zbigniew Mikulski made, and I'll introduce him shortly. So each of these little dots is a bacteria. They form filaments. And here's a scanning electron micrograph from the literature. So what we found in mice that, and I show a picture of Zbigniew here, what we found is that in mice that have our single gene deleted, there are more of these segmented filamentous bacteria, which look like green threads. The red is just showing you the structure of these intestinal villi, and this is a merge or overlay. So the controls have some, but these epithelial cells that lack one gene have much more of these things. I kind of think of them as hair, and the epithelium needs kind of a haircut to get rid of the segmented filamentous bacteria. Well, so what? Well, it turns out that just because of this lack of one gene, inability to make a protein in this border cell type, there were many consequences. And those consequences include changes in the border cells or the epithelial cells themselves, changes in immune cell populations in the intestine, and importantly, increased inflammation, and at the same time, a reduced ability to survive bacterial infections. And you can see that here with a salmonella infection. And the mice that had the gene deleted are incapable of surviving an infection that the control mice survive. I'll just give you another example. And this example also involves some gene surgery, but I'm not going to concentrate that on here. And this is work from Hilda Scharutra's lab. And what she's shown is that your T cells pay attention to your food. And this has to do with the idea, again, that your immune system is monitoring what's going on in the outside world. It's not ignoring it. It's not just an impermeable barrier. What the Scharutra lab did here is they fed mice egg protein. Well, albumin is simply egg protein. And what they measured weeks later was the number of egg protein reactive T cells. So the white bars show the control mice that have a typical diet with diverse proteins, and the black bars show the increase of food reactive T cells. So your immune system is aware of what you're eating. And what happened in these animals is they made two kinds of cells, the red ones and the green ones, and the names don't really matter that much. But these cells prevent you from responding in an inflammatory way to your food. The red ones actually live in the epithelial layer, and the green ones live below the epithelial layer. But these cells make it so when you eat something, be it egg protein or peanuts or whatever, that you don't get diarrhea, you don't throw up, and you don't go into anaphylactic shock, which can kill you within minutes. So being able to sense the outside world and respond to it appropriately is a very important aspect of what the barrier immunity must do. None of this work could be done without highly skilled core facilities that have supreme technical expertise that we rely on for some of our experimental work. And one of the key research cores in the Institute is our microscopy core. And I'm going to, for live in the lab, we're going to go to our microscopy core. And I want to begin that part of the talk by introducing Zbigniew Mikulski. He's originally from Poland. He got his Ph.D. at the Justus Liebig University in Germany. He joined us in 2016 initially as a postdoctoral fellow. He's a brilliant teacher. He's a brilliant technologist. I've written 6 papers with him myself, and more than half of our laboratories have published with Zbigniew, as well as many labs at UCSD. So I'm going to hand the presentation over, and we'll go into the imaging suite with Zbigniew.
Thank you, Mitch. Thank you for the kind introduction. So we are in the BSL2 area. That stands for Biosafety Level 2. That's why I'm gowned like this. And normally I would be wearing a mask, but we have sterilized this room not so long ago, and it's perfectly safe for me to be in this condition. So we are looking at our fluorescence microscope. This is one of our more advanced machines that allows us to do live cell imaging. And let me tell you a little bit about the process of fluorescence. So what I have here is some tonic. No gene was added. And if I shine a UV light on it, I'm going to observe this brilliant blue fluorescence. I hope you can see it on your screens as well. So we use this kind of process where, as a scientific community, as a humanity, we learned essentially to jog the electrons in atoms and make them jump. And when they jump, they produce light of a different wavelength. So we can use this in our microscopes where we can illuminate the sample with different colors of light. And then we have specific probes. The sample preparation is an art, and it's been perfected by members of my team, as well as all of the postdocs that work with us. They're really amazing people to work with, and I very much like being in this lab and in this role, because my greatest satisfaction comes from those interactions where people are just learning how to do stuff. They have no idea how imaging works, and very shortly after getting introduced to that, they fell in love in microscopy, and they start to produce beautiful images. And I'm gonna now switch my screen to share with you a view from the microscope. And I'm going to explain a couple of things that we can do with that. So what Mitch was showing you is that we can observe those segmented filamentous bacteria. Let me just get my screen share going. And this is one of the views of segmented filamentous bacteria that we are able to capture on our microscope. So perhaps on your screens, you will be able to see that those green filaments are actually composed of tiny little segments. And that's where the name of this bacterium is derived. In the red color, we have the microvilli. Those are the pieces of our intestine. In this case, this is mouse tissue. And in the blue color, we see individual nuclei of cells of the barrier. So this microscope is here because around 16 years ago, a private donor essentially changed the way how we do science. Through a private donation, we got a microscope that allowed us to essentially start to create new generation of scientists in our lab. And let me show you a couple of highlights from this microscope. So because it's a microscope that allows us to do live cell imaging, we were able to show how T cells kill insulin-producing cells in type 1 diabetes, how monocytes can literally take a bite out of a cancer cell and they can take it to the lymph node and show it to other cells to mount an effective immune response, or how our very talented colleague, Dr. Sarah McArdle, was able to use this microscope as a platform to engineer her own device that allowed her to record movement of different kinds of macrophages. Those are immune cells in the wall of the artery. And thanks to that, Sarah got her Chan Zuckerberg Initiative Imaging Scientist Award and part of the lab funding comes from that foundation. So in relation to what Mitch was talking about the epithelial function, epithelial barrier, I would like to show you a little movie that we acquired not so long ago. That movie is showing you how the cells behave at the barrier. And they're running around a lot. Let me just find my way around that screen share. Here we go. So what we're looking at is a single plane taken from a three-dimensional image. This is mouse intestine. So I can move you through different levels of that tissue. We're going deeper now, and now we're gonna go back to a little bit more shallow spot. Cells that are bluish, those are the epithelial cells, those are the cells of the barrier, and cells that are green and yellow, those are the immune cells. And the big impact that this microscope has is coming from the fact that we can record those live movies of tissues, and we can observe the behavior of cells. And sometimes you see something that's relatively unexpected, such as that this cell right here in this corner of the screen, it's actually poking through the epithelial layer, perhaps to sample what's on the other side. So looking at the behavior of those cells is really a lot of fun. It's also allowing us to put a much better model of what we're working with. We typically work with cells that are dissociated from each other. That allows us to more precise analyze them. But microscopy is so helpful to put all of this back together and see how those models, how those tissues really work in concert. All of the cells of the immune system talk to everybody else. And what fascinates me is to try to understand how they communicate and how we can affect that communication to help with some of the diseases that plague us. Thank you.
Okay, thank you, Zbigniew. So let me. Okay, thank you, Zbigniew. That was great. I want to make one point as well that the images are pretty pictures, but we quantify everything. So images are not just pretty pictures. Images are data, and it allows us to understand what's going on at a deeper level. The gene that we knocked out in the border cells prevented the cells from moving around in that healthy way with all those consequences I mentioned. I want to talk about a different kind of work for a few more minutes. I want to talk about studies of inflammatory bowel disease patients. We go away from model systems, and in particular, sequencing the genes that are working, that are expressed to make proteins by RNA-seq in T lymphocytes from colon biopsies of patients. And this work couldn't be done without my colleague and friend Vijay and the input also from Christian Ottensmeyer in the United Kingdom. So we started a collaborative study with Southampton in the UK, and we got samples from ulcerative colitis patients as well as healthy controls. And we sequenced the RNA, which is basically getting a read on the gene activity of two types of T cells. And in total, 14,000 cells were analyzed. And this is a study that's still in progress. So here's a representation by a uniform manifold approximation and projection of all the T cells in this study. The CD4 cells are on the left and the CD8 on the right. Each dot is an individual cell. And we have, from each cell, we have thousands of genes that have been sequenced. But two dots are closer to one another if the total gene activity in cell A is close to cell B. And if they're farther apart, then they're different. So we look at the clusters of cells that we observed. This is the total. This includes samples from patients and samples from the healthy controls. From the patients, we have inflamed areas and we have uninflamed areas. Remember, in some cases, the inflammation skips. And we also have samples from patients who are apparently in remission, who haven't had disease flares for several months. And this allows us to understand the pathogenesis of disease in a much better way. So here, again, I'm showing you these UMAP plots, these dots of the cells, and I separated out, instead of showing you all the cells, I separated them. So in the upper left, we have cells that were from inflamed tissue, and then on the right, we have non-involved tissue from the same patients. Here are the remission in the lower right, and here are the healthy controls. And I think what you can see is that we drew a circle around some of these pink and green cells. I think what you can appreciate is that these populations are associated with disease and even present in remission, but almost absent in the healthy controls. So these, we think, are features of the gene expression in those cells that are associated with disease. And the next slide, it's the same representation, but we drew circles around things that are associated with inflammation itself. So these ochre and pinkish-looking cells, color-coded that way, are almost exclusively present in the inflamed areas and absent elsewhere. And we can then sift through this kind of genetic information, and it allows us to identify biomarkers, things that are associated with disease, or that gene expression that might identify individuals at risk for disease. And it also is giving us and others who are doing similar work targets for the development of new drugs. And this is extremely powerful technology. However, it's obvious that everything we find out from this kind of study is correlative. And we need to go back to our model systems to see if we can show cause. And that's why the genetic surgeries I talked to you about in the animal models are important for making additional progress in this area. We come to work every day and we do imagine life without disease. And I hope you've enjoyed this live from the lab webinar. And I hope I gave you some useful and entertaining and interesting information about barrier immunity. Thank you. And I'll hand it back to Erica.
That was so much fun, Mitch. Thanks so much. We have some fantastic questions now from our audience and our friends and supporters. And while you're listening, you can write in more as well. I'll start moderating the discussion with Mitch and Z. So you've shown us about all the bacteria in our guts. And of course, we hear so much about the microbiome and how it's influenced by our food, our pets, our travels. One of the questions is that good or is that bad?
That's a very deep and important question. There are microbes that are thought to be pro-inflammatory, and there are microbes that are thought to be more healing and beneficial. And we know that, for example, people with inflammatory disease have what they call dysbiosis. They have outgrowths, and they tend to have more of the inflammatory bacteria. The problem is that these communities are so complicated. Think of it as a little town with 500 or 1,000 people. And it's not a simple thing as if, well, there's one bacteria, we get rid of that, and you're okay. Unfortunately, probiotic cures for inflammatory bowel disease have not been extremely successful. There are some studies where they show some benefit and others where they haven't. And so I think we're going to have to, if we're going to use the microbiome as basically a therapeutic window, I think we have to understand the stuff. Remember that very complicated slide, the metabolites, and not be so worried about, what particular, whether you have more proteobacteria, you have more Bacteroides, or you have something else with an unpronounceable name, but what the overall metabolic input is, and can we manipulate that?
You mentioned,
generally speaking, diversity is better than having to be monocolonized by a single organism.
Truly.
But it is very complicated. And the old saying about, having diversified diet and having physical activity definitely still works. It's very unspecific, but we're just at the beginning of understanding how to manipulate that.
Yeah, I agree. And also, if you have an inflammatory disease and you have a diet that you feel good with, it makes you feel good, stick with it.
Absolutely.
Of course, that's obvious.
I see a therapeutic question for Mitch and then an equipment question for Z. So linking together a couple of therapeutic questions. Mitch, you just mentioned IBS and you've researched colitis. Now, some of the people in the audience are wondering about influence of cancer treatments on gut inflammation when there are immune-related adverse effects. And also there's another person writing in that has lymphoma and IBS at the same time. Are these related? How can treatments influence inflammation at your mucosal barriers? What do we do about that?
Yeah, so there are a number of studies that show that there are people who respond well to checkpoint blockade, for example, the latest, greatest, if you will, immune therapy for cancers, many kinds of solid cancers, melanomas, lung cancer, and so on. There are people who respond and people who don't respond. And a number of groups have looked at the microbiome of responders and non-responders, and they have made correlations with certain bacteria. And in fact, in 2018, there were 3 back-to-back papers in a very prestigious journal, Science. And each of the groups found a bacteria that correlated with lack of responsiveness, and they each found a different bacteria, which to me comes back to the diversity question. And remember, these are correlative studies. And so I think what we need to do to understand that is go deeper and understand the metabolomic footprint. What are these bacteria making? So it's not just that Akkermansia, for example, which is the name of one of them, is if you have too much of that, you're not going to do well with checkpoint blockade. It's probably more a matter of what your communities are making that might make you, and that might contribute, and they're not the sole factor, but that might contribute to a better outcome in cancer immune therapy. And the second question that you asked was about whether having a leukemia, I think it was, and IBD are related. Not necessarily, but what I can tell you is that if you have chronic inflammation in the intestine, your risk of colon adenocarcinoma is greatly, greatly increased. So a well-controlled inflammatory response is good, but a chronic one with all the oxidative stress and things going on can be a contributing cause to cancer. But I believe with the information I have, I wouldn't believe that the leukemia and the inflammatory bowel disease are related.
Z, an equipment question. So you talked about the gift that keeps on giving and how that instrument gave us so much rich data to understand so many different diseases. How much was that 2-photon microscope? Could the donation cover the entire cost? And where are we now? How old is that instrument?
Sure.
Could you do better studies with something new?
That's always the case. The instrument itself is 16 years old.
16.
16. We got it in 2007, and the purchase price of the bundle of the 2-photon laser, and ask me more questions about this because that technology is absolutely awesome, and the microscope was $525,000. That was in 2007 dollars, and that amount of money would not be sufficient these days to replace the system. So in about 10 years after our original purchase, we upgraded our microscope to the newer generation model. And that was mostly driven by the fact that computers got so much better in that time, and the detectors got so much better that it was, our old system was really starting to show its age. So one of the reasons why this microscopy table on which the system is sitting is so big is because we were really hoping to one day be able to afford to upgrade our laser that's 16 years old.
because that laser is still a marvelous piece of technology, but of course, progress has been enormous in that space as well. And one of the interesting features of the newer generation of lasers is that they can shine two different wavelengths of light. So we can observe two different populations of cells at the very same time. If you remember, when I was showing you operation of the microscope, I was switching the light from one color to the other. And that's fine for a lot of studies. But when we're trying to see how two different cells interact with each other, having that kind of a laser source is fantastic. And the cost of a new 2-photon laser is about $200,000, with a $100,000 discount that we got because we negotiated that down.
And what more could you do or see if you had that see?
So it's seeing more colors at the same time, and that instant interaction is very important. So, for example, we study here how inflammatory cells are recruited to tissues, and they move very, very, very quickly. So having the ability to image two different colors at the very same time would be critical for that work and would really help us to better understand it. Right now, we're limited to one, essentially. And the other thing is that those new generations of lasers are allowing us to see more deeply into the tissue. Right now, depending on the tissue of interest, we're limited to anywhere between 100 and, let's say, 200 microns, which translates roughly to between 10 to 20 cell layers. And a lot of the interesting things we want to study is essentially happening deeper.
How deep could the new technology let you go?
So there is what we're using currently is 2-photon microscopy, but with the newer technology, we can go with 3-photon microscopy. And in certain tissues with good conditions, you can go 700 microns down. So we have some studies that right now are looking at neuroinflammation and Alzheimer's disease, neurodegenerative diseases. And that kind of instrumentation would allow us to actually look deep into the tissue to the level in the brain where those changes are occurring.
In the brain. What does 2-photon or 3-photon mean, Z?
Yeah, so in normal everyday existence, even if we have something that is fluorescent, what we are really observing is a single photon process. So we can imagine this as a ball that hits something, and that something gets excited and it's throwing another ball of a different color. And that is a single photon process. 2-photon process happens when two balls at exactly the same time hit exactly the same target. And we are talking about photons, very, 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 very small things. So in order to get that to work, we need to use incredible technology that's going to compress and pack a lot of those tiny balls into a packet, a pulse. This laser is a pulsed laser, and it works on a scale of time that's kind of unimaginable to us. So it works in femtoseconds. One pulse takes about 200 femtoseconds. So I'm going to take it to a scale that we understand and know. If I compare the width of the pulse of that laser to 1 second, then 1 second would be stretched to 440,000 years. Okay, it's absolutely bonkers. And the 3-photon process packs even more juice. And now you have 3 photons interacting with this. And here's why it's important. It's important because when you add 2 photons together, you can use photons of lower energy that are less damaging to the tissue, and that can also travel more deep inside our bodies. So what I'm showing you right now is I'm showing my cell phone that shines light through my hand. And the only color that you can see there is reddish. And that's because red light of lower energy, of longer wavelength, is less absorbed by our bodies. And that's why 2-photon microscopy is so popular for studying immune cells deep in tissues.
Fantastic. Mitch, there's a lot of questions in the chat about different kinds of therapeutics and also bridging different kinds of data. So one that's interesting is we looked at the microscopy data of Z, and they've also talked about the RNA-seq data and all the other kinds of data and bridging all that kinds of information. Carol asks, do you have RNA-seq data from patients treated with anti-TNF or other biologics, and what's the impact?
Yeah, the study, these patients were fairly, were naive mostly for biologics. But other people are certainly doing those kinds of studies. And yeah, I think the work we're doing, I know the work we're doing is right at the edge, but we can't do everything at once. So the way our study was designed was to see, really look and see what might be underlying the immune system when there is disease, even if you're not in the inflamed area. And then what gets you to the next level? Because I think we want to try to treat both. We want to try to cool down the inflammation, but then we want to make an underlying change so that the gut or any other tissue, in fact, if it's working in a different system, so that the tissue returns to more its normal state. But there is a lot of work going on now as well with people treated with different biologics. And what's really encouraging is some new ones coming along. And I certainly, I do some consulting for companies that are developing new ones. And I see very promising studies. None of these are cures. And they, like the checkpoint blockade, they're usually treatments that work in some people and not others. And we need to be more sophisticated about understanding why that is. What are the differences?
So, another aspect of the power of the immune system is when it goes wrong, and autoimmunity. And some of our audience members are writing and saying, why is it that some people get autoimmunity and not others? And why do some people have multiple autoimmune disorders at the same time?
Yeah. Well, autoimmunity is a difficult topic. So we know there are genetic risks and genes contribute, but they're not the kind of genetic risks where if you take this one gene, you have the disease, right? If you think of sickle cell disease or Huntington's disease, if you have a mutation in those genes, you have that disease. And in autoimmunity, it's more the action of variation, and there's normal human variation in our genes, variation in several genes contributing to genetic risk. But genetic risk is not everything. And the environment is very important. One way that we can demonstrate that is identical twins don't, if one twin has, let's say, Crohn's disease or type 1 diabetes, the other twin doesn't necessarily have it. So clearly the environment is very important. And so it's really multifactorial. We do know that, and part of the environment, of course, is the microbiome, but it's also the diet and it's stress and other things that you might be exposed to. One thing that's really evident is that a lot of these diseases are increasing in prevalence. Some of that could be bias of diagnostics, but clearly, even if you allow for that, a lot of diseases, think of food allergies, right? We used to, when I was growing up, nobody thought about, not bringing peanuts to the Little League game for the kids to eat. And now it's very different. And that's something even that we really don't fully understand. We know there are environmental causes of the increased risk. There's an idea that perhaps we're too hygienic, that we're too clean. We're not exposed to enough parasites. Well, I don't really want to have whipworms in my gut, but yeah, it's possible that there's that hygiene is an element of it. There are other theories about changes in our diet with processed foods and other things. We really don't understand even something so basic as to why so many autoimmune diseases are increasing. But there's a lot of really brilliant studies going on, and I'm sure there'll be more and more progress as we get better understanding of human genetic individuality, as in the kind of studies that I showed you from the patients.
So those kinds of large data multifactorial studies can illuminate why one person gets sick and another one doesn't, what the actual explanation is.
Yeah, truly, I agree.
Now, some of our audience members are asking about pediatric disease specifically. Can you comment on some of the areas of pediatric disease studied at LJI? So Farhat I studies pediatric cancer, and of course, Susan Shrestha studies pediatric viral infections like dengue, and Chris Benedict cytomegalovirus.
Yeah. Well,
it's involved in looking at eosinophilic esophagitis. And this week we had a meeting to discuss how we can help to look at what's going on in these tissues with our imaging tools. So there's a lot of activity around LJI about this topic, but Mitch knows a lot more about this than I.
No, I think you both of you have encapsulated it very well. The pediatric cases, I mean, the esophagitis that Zbigniew talked about affects mostly boys, and it's a tough disease to treat. There are pediatric versions of diseases like inflammatory bowel disease, and they're usually more severe, or pediatric versions of arthritis. Those diseases are often more severe and often have, well, not often, but in some cases do have better defined genetic lesions than the typical case onset when you're 30 years old or whatever your age is.
So more questions are coming in on that topic of the nature versus nurture in autoimmune disease. And, you know, I remember the old studies, so when they looked at identical twins, so the same nature versus non-identical twins, the same nurture, right, the same environment. Decades ago, the stronger link for autoimmune disease was in the identical twins. And now with the rise of autoimmune disease in the last 20 years ago, and especially among girls and women, we see a stronger link with the non-identical twins, so environmental. Some of our audience members are asking for a little more information on that. Can viruses trigger autoimmune disease, or is it an environmental toxin? Is it something about our food and the way our food is grown, or is it our more limited sunlight exposure that we have now than decades ago?
Yeah. Well, all of those things have been proposed, and none of them have been proven. I come back to this hygiene idea because to me, it almost seems like it's not falsifiable. Therefore, it wouldn't be a scientific theory almost, because, there's always a question, well, how hygienic? And for example, we know with asthma that, your risk of asthma decreases if you have pets and maybe the dog or cat dander or the microbiota of the animals is helping in some way. But those effects are not huge. And then there's a very interesting study where they were comparing the Amish to the Mennonites, which both have an agricultural lifestyle, but the Amish had a lower rate of asthma, and the difference was the Mennonites have mechanized farms, and the Amish are still horse and buggy. So it's not simply enough to live on a farm and be exposed to all the microbiota of the animals or whatever, but you have to live on an Amish farm to really reduce your risk. So I think the effect sizes are just not big enough to know for certain. There is evidence in type 1 diabetes, for example, of enteroviruses being a potential cause, and that's a viable theory, but no one has the real proof that a particular enterovirus, intestinal virus, or Coxsackie virus, which has been a candidate, no one has proof that those are really strong contributors to disease.
Lots still to discover.
And just a comment on that, we call them autoimmune diseases, but in reality, this is a variety of very different disease processes that are just lumped together because we lack the understanding of what's actually going on. In some of them, there is a process where our own body starts to recognize our own antigens. And that often happens when there is a lot of neutrophils that are being recruited to areas of chronic inflammation. And then antigens from those neutrophils are being seen by the immune system. But there's so many different ways how this can go wrong. So it almost looks like initially when we were thinking cancer, we were thinking tumors, and surgery was the only thing. And then we started to discover that they're actually very heterogeneous, and there is no one fix all for them. And just a comment on the environmental factors, I think would all benefit from cleaner air. Diesel particles and diesel exhaust and things like that. You know, sometimes I'm biking up the Genesee, and if an older classic car is passing me, by the time I'm on the top of the hill, I feel like I have an asthma. So I think this is all very important. Unfortunately, we don't know for sure what's really the causing agent.
Yeah, I think we have to take into account the genetic individuality, that in a way, some of these diseases are syndromes, and to really treat them effectively, we need to consider the genetic individuality and the pathways that might be involved in one person versus another.
That's absolutely right. And that's the direction the Institute is going, is the individuality of your immune system and your own genes and your own exposure. So ladies and gentlemen, as you go and eat your lunch, or maybe you've already been eating your lunch, you can think about how your immune system is sensing it, and your gut immune system is protecting you from the outside world. You've gotten to hear today just from one of our faculty and one of our core experts and one of the areas of research that we do. If you would like to see more, we would love to welcome you. You can come and see the facilities and the microscopes for yourself and talk to our researchers. If you'd like to learn more, you can go to our website, www.lji.org. If you'd like to support this research and be a part of it and make it part of your legacy, you can donate at donate.lji.org/LFL, live from the lab, or by scanning that QR code right there with your mobile phone. I'll leave that up for just a couple of minutes while you get your phone out. Or you can write me or our Vice President of Advancement, Kelsey Dale, at any time to be a part of the kind of research that Mitch and Zbigniew and all of our other scientists are doing. Thank you so much for joining us today. It's always fun to get to talk to you and to learn more about the incredible research that goes on here in our immune systems. Have a wonderful day.